are killed preparations of microorganisms used for immunization. So vaccines are live attenuated or killed preparation of microorganisms used for immunization. What are recombinant vaccine? A very important 2-mark definition. A vaccine produced through recombinant DNA technology, which involves inserting the DNA encoding an antigen, specifically if you see maybe a bacterial or viral surface protein that stimulates an immune response into bacterial or mammalian cells, expressing the antigen in these cells and then purifying it from them. I repeat, recombinant vaccines. A vaccine produced through recombinant DNA technology, which involves inserting the DNA encoding an antigen, such as bacterial or viral surface protein, that stimulates an immune response into bacterial or mammalian cells, expressing the antigen in these cells and then purifying it from them. Simple to make you understand this definition. So there is something called a surface antigen in virus, and of course antigenic determinants in what is called as a bacterial cell. So you will remove the DNA that codes for it, you will clone it in a vector molecule, you will insert it in a non-pathogenic strain and purify it. That is what is a recombinant vaccine. So what are the different types of vaccines? So we have live attenuated vaccine, killed inactivated vaccines, cloned genes of microbiological antigens, purified microbial macromolecules, synthetic peptides, and of course DNA vaccine. So what we will study with rDNA technology is specifically on DNA vaccine. So types of vaccines, I repeat: live attenuated vaccine, killed inactivated vaccine, cloned genes of microbiological antigens, purified microbial macromolecules, synthetic peptides, and of course DNA vaccine. So how was these traditional vaccines produced? So the vaccines which we are studying under recombinant DNA technology, we call it new generation vaccines. Traditional vaccine are those which are produced in the older forms. If you see, the traditional vaccines were obtained by growing the organisms, the microbial cultures in either animals or simple bacterial cultures or maybe egg inoculation. So there was 3 means by which we could produce vaccine, either growing the organism in the animals. For example, Vaccinia virus is grown in calves for smallpox vaccine. Rabbit brains are used to produce rabies vaccine. I repeat, growing in animals. I've given you 2 examples: Vaccinia in calves for smallpox and a rabbit brains for rabies. And of course, simple bacterial culture, you can go in for cultivation of Vibrio cholerae, the cholera vibrio, then you can go in for inactivating or killing them. The next one we have is growing in eggs, and 2 examples I have given you is influenza and Vaccinia virus, and then you can go in for inactivation. I repeat, traditional vaccines can be produced either growing the organism in animals, simple bacterial cultures, or growing it in eggs by means of egg inoculation techniques. And of course, in case of the eggs, if you see, greater than 100 million eggs were used for influenza in the USA every year. So even now, if you see, in case the influenza virus could be cultivated only by growing it in eggs, greater than 100 million eggs are used every year for production of influenza vaccine in USA. So what are the limitations to traditional vaccines? A very important 2-mark question again. Why did we go in for new generation vaccine? What was the limitation to traditional vaccines we could find? So the first limitation we had is we cannot grow all organisms in culture. So all organisms culture la valakka mudiyuma appadinu paatha valakka mudiyadu. Viruses, they cannot be cultivated in an artificial medium. So there are certain bacterial cells even you cannot grow, and you need an animal model for its growth. So you cannot grow all the organisms in culture. Again, if you see another limitation we have is the safety of laboratory personnel. Most of these organisms are pathogenic forms. We are doing what is called as attenuation. Attenuate panna porom. So pathogenic form handle pandranala, we cannot ensure safety of the lab personnel. Of course, very expensive. Again, the reason is because I told you already, greater than 100 million egg is not a joke. So you have to rear hens for that. So you need larger space to cultivate these type of animals, a model that can be used for production of vaccines. So the expense for producing traditional vaccine touches higher rate. And it is when there is insufficient attenuation seriya nadakala appdin sonna, there is always reversion to an infectious state. Or virus particle irukku, adha neenga attenuate pannittu vaccine produce pannanum. And the attenuation சரியா நடக்கல அப்படினா பாடியில நீங்க vaccine போட்ட பிறகு அது it can be reverting back itself to pathogenic form. So வியாதி உண்டு பண்ணக்கூடிய திறமை அதிகமாகிறதுக்கான வாய்ப்பு உண்டு. Then you need what is called as refrigeration, and of course it does not work for all the infectious agent. Again, animal models எல்லாருமே வளர முடியாது. So you cannot work it on for all infectious agents. And of course, the final one is the one who are going to receive maximum vaccines. If you see immunization schedule, second year immunology. So the problem for us, if you see, is most of the infants or children are going to receive them. So they will have immature immunity. In that case, continuously when you administer a booster dose or a vaccine that can be reverting itself, it is going to harm the child that receives it. So the limitations to traditional vaccines, I repeat, can't grow all organisms in culture. The safety to laboratory personnel can never be ensured. It is expensive. Insufficient attenuation may lead to reversion to infectious stage. It requires refrigeration. Do not work for all infectious agents, and infants, children will receive them and they will have immature immunity. So to overcome all these limitations and disadvantage, we came across with what is called as new generation vaccines, the recombinant DNA vaccines. So the new generation vaccine, the recombinant vaccine. So recombinant DNA technology is being used to produce new generation of vaccines. What probably is carried out, if you see, the virulence gene are deleted. Even though the virulence gene are deleted, the organism will have surface antigen that can stimulate an immune response. They may not remain pathogenic. They will induce immune response instead, but it cannot revert itself to what is called as pathogenic form because you are removing its virulence gene. So virulence genes are deleted. An organism is still able to stimulate an immune response. So you can use live non-pathogenic strain to carry antigenic determinants from pathogenic strain. For example, hepatitis B surface antigen, if you see, the host in which you will use it, if you see, is the Saccharomyces cerevisiae, which is an yeast cell, and they are harmless cell. They are non-pathogenic in nature. Hepatitis B surface antigen or vector kulla potu adu yeast cell la clone panni, you will produce HBsAg vaccine. So yeast cell under it is a non-pathogenic form. So definitely we can ensure safety of the personnel who are receiving the vaccine. If the agent cannot be maintained in culture, so I already told you in previous slide, you cannot grow all the organisms in an artificial medium or a culture. So in that case, genes of proteins for antigenic determinants can be cloned and expressed in an alternative normal flora, the host example, Escherichia coli. So I repeat the slide, new generation vaccines. Recombinant DNA technology is being used to produce a new generation of vaccines. Virulence genes are deleted. An organism is still able to stimulate an immune response. Live non-pathogenic strains can carry antigenic determinants from pathogenic strains. If the agent cannot be maintained in culture, genes of proteins for the antigenic determinants can be cloned and expressed in an alternative host example, Escherichia coli. So predominantly genetic engineering is used to produce two different types of vaccines, what you call as DNA vaccines, the recombinant vaccine, protein produced by genetic engineering. That is why they are also referred to as protein engineering. Express protective antigen is safe and easy to grow the organism. So two such examples I have given you is the hepatitis B vaccine and human papilloma virus vaccine. So hepatitis B surface antigen expressed in yeast and papilloma L1 antigen expressed in yeast. So what are the advantage and disadvantage of recombinant vaccine? So everything will have an advantage and disadvantage. So advantage of recombinant vaccine, if you see, they are safe because we grow the organism's antigen in a non-pathogenic yeast strain. So they are safe, they ensure safety. Number two is it is easier for production. So in case of difficult to grow virus like hepatitis B and virus and human papilloma virus, viruses requires a live human cell for its growth in tissue culture. It requires an animal model for its growth. But in case if you can produce recombinant vaccine, it is a very easier mean. You can isolate the DNA, coding for antigen, you can clone it in a non-pathogenic strain. So it is easier for growing the viruses, of course, like hepatitis B, HPV, for production of recombinant vaccine. And disadvantage, if you see, of the recombinant vaccine, the first disadvantage you will see is you have to go in for identifying the protective antigen. So antigen, viral antigen or surface antigen are larger in number. So you need to identify the antigen first that gives protection to human beings. So identification is the first part that you cannot call it disadvantage, but you need to work a lot for this. So need to identify protective antigen, obtain antigen in correct configuration and conformation. Antigen மாத்தி ஏதாவது எடுத்தோ இல்ல மாத்தி அமைச்சு clone பண்ணீங்க அப்படினாலோ it is not going to play with somebody else. It is going to play with human lives. It is going to play with the lives. So definitely correct confirmation is required in order to avoid contamination and avoid detrimental effect of the vaccine. And of course, these recombinant form of vaccines will induce poor cell-mediated immunity, and definitely it requires adjuvants for its function. So adjuvants கொடுக்க வேண்டியதா இருக்கும். I repeat the slide. The advantage of recombinant vaccine, safe. So it can be easily grown in non-pathogenic yeast cell, easier for its cultivation, for example, hepatitis B and human papilloma virus. And disadvantage, if you see, you need to identify protective antigens, obtaining antigen in correct conformation, and poor cell-mediated immunity, and it requires adjuvants to function. So we are stepping into hepatitis B, the recombinant vaccine, hepatitis B surface antigen. This is an introduction slide of hepatitis B. More than 2 billion people alive have at some time been infected with hepatitis B virus. Around 350,000 remain chronically infected carriers. So the larger amount of people are the carriers. Carriers are those individuals who will harbor the organism without any ill effect by the organism. So their only ambition or their only function is to transfer what is called as infection. So around 350,000 people remain chronically infected carriers. Every year, per year, if you see, every year, there are 4 million acute cases and 1 million deaths because of hepatitis B. And the carriers usually, if you see, 25% of the carriers are children less than 7 years, and around 10% of older children and adults become carriers. So the ratio of carriers, if you see, are more with children. There are 25% of children who always remain as carriers below 7 years of age. I repeat the slide. More than 2 billion people alive have at time been infected with hepatitis B virus. 350,000 remain chronically infected carriers. Every year, there are 4 million acute cases, 1 million deaths, and carriers, of course, as we see, 25% of the children who are lesser than 7 years become carriers, and 10% of the older children and adults become carriers. Now, regarding the structure of hepatitis B virus. It is an enveloped virus that has double-stranded DNA molecule as its genetic material. So if you see the diagram, it shows you it has an envelope, it is an enveloped virus, and you have a core structure. Core is internal structure, and it is a double-stranded DNA virus with one strand being open, single-stranded DNA at an end. And if you see, you have two different type of antigen. Hepatitis B surface antigen, which is written or designated in the picture as HBsAg, and you have something called as core antigen, hepatitis B CAG, HBsAg, and of course we have pre-S entity, pre-surface entity. And this particle is called as infectious Dane particle in the structure. Okay, so I repeat the structure. Hepatitis B virus is an enveloped virus, and the core structure, it will have double-stranded DNA molecule as its genetic material, hepatitis surface antigen, and of course hepatitis B core antigen. So what we are going to do is we are going to remove this DNA coding for hepatitis B surface antigen and produce a vaccine in an yeast cell. During infection, large amounts of hepatitis B surface antigens are produced. Only small amounts of hepatitis B surface antigen combine with the cores to form whole virus. So HBsAg, HBsCg, when it combines, it becomes an infectious virus particle. So remainder is released into the bloodstreams as spherical particles or filaments. Antibody to HBsAg indicates clinical recovery and is associated with immunity to hepatitis B. So in the previous picture, I told you two different types of antigens are there. Number one is hepatitis B surface antigen, hepatitis B core antigen. When hepatitis B surface antigen combines with hepatitis B core antigen, the particle becomes infectious. So viral infectious particle is produced to cause what is called as hepatitis infection. In case of the hepatitis B surface antigen, most of them do not combine with the core structure. They are released in bloodstream. In case hepatitis B surface antigen in a larger amount is supplied to the bloodstream, it indicates very clearly that the hepatitis B surface antigen is inducing clinical recovery because it is not combined with core antigen, and that is the reason why we are removing these hepatitis B surface antigen, isolating the DNA responsible for it, and then we are going to clone it into the yeast cell. I repeat this slide. During infection, large amounts of hepatitis B surface antigens are produced. Only small amounts of hepatitis B surface antigen combine with the cores to form whole virus. Remainder is released into the bloodstreams as spherical particles or filaments. Antibody to hepatitis B surface antigen indicates clinical recovery and is associated with immunity to hepatitis B. So what is the aim of hepatitis B vaccination? So why do we go in for hepatitis B vaccination? If you see, as I told you in the previous history slide, there are so many people who remain carriers, so many cases of infection every year. So the main aim, if you see, is to reduce clinical disease. So disease reduction and transmission of infection from carriers, and to reduce hepatitis that leads to what is called as liver damage and liver cancer. So I repeat, vaccination aims at reduction of clinical disease, hepatitis B virus transmission, and of course chronic hepatitis and its associated liver damage and liver cancer are the aim of hepatitis B vaccination. So this picture very clearly tells you how are these recombinant subunit vaccines are produced for hepatitis virus, that is preparation of genetically engineered vaccine. So you have hepatitis B virus on the left hand side with DNA depicted or circulated in orange color, which is isolated surface antigen gene. So you are isolating surface antigen gene, that is HBsAg coding gene from hepatitis virus, you are inserting it into yeast cell. Of course, for this case, you should use an yeast vector. You can use an yeast artificial plasmid or yeast integrative plasmid, anyone that can hold the size of the antigen. So you're inserting the vector inside the modified yeast cell so that it can produce hepatitis B surface antigen. You're growing the organism in pure culture form, the Saccharomyces cerevisiae, the yeast cell is grown in pure culture form, and you can purify the vaccine for human trial or for substituting to human beings as a preventive methodology. So vaccines are used for prophylactic measure, so you can manufacture vaccine after its purification. I repeat, hepatitis B surface surface antigen. The gene coding for hepatitis B surface antigen is isolated. It is inserted into an yeast cell. The modified yeast cells produces hepatitis B surface antigen. It is cultivated in pure form to produce HBsAg, and the vaccine is purified for manufacture. So this is how there is preparation of genetically engineered vaccine. It is not only applicable to hepatitis B virus vaccine, any genetically engineered vaccine, of course, the main requisite is the DNA responsible for what is called as its antigen. So antigen code pannakuriya gene isolate panni enna possible vector irukko adula insert panni, you're growing it in a non-pathogenic form of strain. In case you're using Escherichia coli is a normal colon bacilli. It is a normal flora of your intestine. So again, non-pathogenic form, you can very well isolate from the colon and you can use for the production of vaccine. But for the production of recombinant subunit vaccine, we employ for the purpose of easiness the Saccharomyces cerevisiae, which is an yeast cell. So this is the description. The gene coding for hepatitis B surface antigen was discovered in the year 1970. The gene is isolated and inserted into an yeast cell. As the yeast cell grows, it produces large amounts of hepatitis B surface antigen. The HBsAg thus produced is extracted and purified for injection as a subunit vaccine. Advantages of hepatitis B surface antigen recombinant vaccine. So main advantage, if you see, is they can be produced very quickly, produced more quickly, produced in larger quantities. So or conical flask or a test tube, you do not require animal models, big areas for its growth. So you can produce them in a very large quantities. And of course, since the surface antigens gene is only removed and cloned, it is free from infectious virus particle because it is no way associated with the core antigen surface. So advantages of hepatitis B surface antigen recombinant vaccine, I repeat, produced more quickly, produced in larger quantities, and free from infectious virus particles.